We're here at the CROI 2019 here in Seattle. We're here with Gary Maartens from the University of Cape Town, and we appreciate you taking the time to visit with us. We just got out of a press conference, and you gave some pretty interesting data. And I'm going to let you describe the presentation you made, and then we'll talk a little bit about it.
Sure. Okay, so there are two new TB drugs, which are the first two TB drugs to be licensed in more than 50 years, more like 45 years, I guess. And they are drugs from completely different classes. So therefore they could be used in drug-resistant TB. Both drugs are registered, one not yet in the US, the other is registered only in the European Union for now. But they are recommended by the WHO for the treatment of drug-resistant TB. The problem is that they will therefore be used in combination, and they have a potential shared side effect in that both of them do a cardiac abnormality, which is the conducting system of the heart. They prolong something called the QT interval on the electrocardiogram, and that is a potentially dangerous side effect because if that happens in a severe way, it can result in a life-threatening rhythm disturbance of the heart.
Arrhythmia. Yeah.
Correct. So our study was to see whether combining these two drugs would be safe. And when you combine different drugs that prolong the QT together, their effect can be more than additive. And we wanted to make sure that that was not the case. We knew more or less what their QT effects were as single drugs from phase 2 studies that the companies had done.
So when you would combine them, you would do half the dose you would normally use on each of them? Is that?
No, we used full dose.
Full dose on each of them, but yet you still had only, so additive meaning doubling?
Well,
because of the dose?
Additive would be the QT effect of the one drug would add to the QT effect of the other drug, or would it be more than additive?
Right.
So they don't necessarily have exactly the same QT effects.
I got you.
So we did a randomized trial which was funded by the ACTG of people with drug-resistant TB who were on therapy. They had to be on therapy for a minimum of a week and a maximum of 2 months. And they were randomized to get bedaquiline added to their therapy, or this other drug, delaminid, or the combination of bedaquiline and delaminid in the third arm. And then we looked at the effects on the ECG frequently during the study. Both drugs have a slow onset of the QT effect, which takes weeks to months to develop and to plateau, rather than with other drugs, which happens very quickly. And we were able to show that the effect was indeed no more than additive. No patient in the study got severe QT prolongation. There were only some mild grade 1 or grade 2 QT prolongation events.
But you could confirm that it was actually from this drug?
Correct. We got almost exactly an additive effect. So the QT effect of the one drug was just under 12 milliseconds, from the other drug was just under 9 milliseconds, and the two together was just under 21 milliseconds. So it was exactly additive. And 21 milliseconds is not a large effect. But the caveat is that this is in a controlled environment where we did a baseline ECG, and one is a little bit concerned that in the field, these drugs will be combined, often with other drugs that can also cause the QT, and the patients might not be so well screened. But we can conclude from our study that there is a modest QT effect, which is additive when you combine these two drugs. And we think that can be managed in the field.
So people who were tested, they were able to take, in the study, were able to take whatever drugs they needed to take, but they would be recorded as having been added to their regimen, whether it be like for cardiovascular issues or whatever it is, whatever other drugs they might be taking.
There was a list of drugs which were prohibited.
Okay.
So we tried as far as possible to remove any drug that would have a QT effect of its own, or any drug that would affect the metabolism of these two investigational drugs. So as is usual in a study like this, there's quite a long list of prohibited medication.
But so where do you see this going? Are they going to be testing? They have to be tested on down the line every so often, just to make sure this is not the case and that they're not having this issue.
Under ideal circumstances, because these two drugs and a number of other drugs that are used in drug-resistant TB all have this shared side effect. So under ideal circumstances, what you would want to do is a baseline ECG to make sure there wasn't a problem at baseline with a long QT. And then you would want to, at intervals, repeat the ECG just to make sure that the QT effect in an individual isn't much more than you would expect on the basis of
This would be
research.
for treatment after its approval and so
Correct,
forth.
yeah. As I said, both drugs are already approved and are beginning to be used together as recommended by the WHO.
So where do you see, what are the next steps on this to ensure that it's a modest effect, but it still is there? And what do you see the next steps would be?
Yeah, I think modest QT effects by themselves aren't terribly alarming, but if it were, for instance, a mild disease, for instance, hay fever, there was a drug with a very modest QT effect, which was withdrawn from the market some years ago because there were deaths due to this arrhythmia. Now, that was a very rare event, and in the context of a very severe disease like drug-resistant TB, that risk is acceptable. So it is an acceptable risk. Where I think the field is going in future is we need to do careful observational studies when other QT prolonging drugs are added to this combination.
So you have to be watchful of what drugs you add and have, I mean, it is polypharmacy. It's really concerning because people have a lot of other things going on that they have to treat for. So we need vigilant doctors. Maybe that's the moment to take that, to tell doctors they have to watch for, you know, polypharmacy. I know that a lot of even some of the clinics, you know, basic county clinics, have pharmacists who kind of review every time the person comes in, review any new drugs, if there's any interactions, because it is important to do that.
Yeah, that's ideal, but in many low-income country settings, that doesn't really happen. So typically for drug-resistant TB, between 5 and 7 drugs are used. Then if you've got HIV, there's another 3 agents that are used, and there are often comorbidities with other drugs added in, and polypharmacy is dangerous, as you point out.
Were there any ethnicity issues in this for one population over another as far as
our study was done in Peru and in South Africa, although most of the patients who were enrolled were from South Africa. But within the South African population, some of the patients were Black African and the others were of what we call colored mixed race population. And the Peru population was somewhat different. So we had reasonable ethnic diversity in the study, but we're not powered to look at any ethnic effects.
That's good. So I very much appreciate your work, and I hope that, well, the presentation was really good and well done, and your press conference was excellent. So I'm excited that we have the opportunity to visit with you.
Thank you.
Thank you very much.
Okay, cheerio.
Thank you.